This video covers bacterial physiology. We will define nutritional types of bacteria, list growth conditions, describe the bacterial growth curve, and state important metabolic pathways. Finally, we'll see how metabolic knowledge aids in bacterial identification. Bacteria can be broadly classified by their nutritional types, specifically how they acquire carbon and energy, often categorized as autotrophs and heterotrophs. Nutritional types include phototrophs, which use sunlight for energy, and chemotrophs, which obtain energy from chemical compounds. Chemotrophs are further divided into chemoorganotrophs, oxidizing organic compounds like most bacteria, and chemolithotrophs, oxidizing inorganic substances such as sulfur-oxidizing bacteria. Bacterial growth is dependent on several key conditions. These include atmospheric conditions like oxygen and carbon dioxide, temperature, pH, and the availability of essential bacterial growth factors. Regarding oxygen, bacteria are classified as obligate aerobes, which require oxygen, microaerophiles needing reduced oxygen levels, facultative anaerobes, growing best with oxygen but capable without it, obligate anaerobes, which cannot grow in oxygen, and aerotolerant anaerobes, which are anaerobic but tolerate oxygen exposure. Carbon dioxide is another atmospheric requirement. Capnophilic organisms, such as Neisseria species, prefer or require increased levels of carbon dioxide for optimal growth. Bacteria are also grouped by their preferred growth temperature. Psychrophiles thrive in low temperatures, mesophiles grow best at moderate temperatures, and thermophiles require high temperatures, like the Thermus aquaticus used in PCR. pH requirements categorize bacteria into acidophiles, growing optimally between pH 0 and 5.5, like Helicobacter pylori. Neutrophiles prefer a pH between 7.2 and 7.6, which includes most pathogenic bacteria. Alkalophiles thrive in high pH, between 8 and 11.5, such as Vibrio cholerae. Bacterial growth factors are organic compounds cells cannot synthesize but need to grow. The three main classes are amino acids for protein synthesis, vitamins needed in small amounts, and purines and pyrimidines for nucleic acid synthesis. Bacterial growth involves increasing size and number. Population growth is studied in liquid culture, where bacterial counts plotted against time yield a growth curve for microbes reproducing by binary fission in a batch culture. The bacterial growth curve begins with the lag phase, where cell numbers remain constant while cells prepare for growth. This is followed by the log or exponential phase, where the cell number increases exponentially at the maximum rate. In the stationary phase, cell division stops due to nutrient depletion and toxic product accumulation. The number of new cells equals the number of dying cells. Finally, the decline phase sees a decrease in population due to cell death, sometimes caused by autolytic enzymes. This diagram illustrates the four phases of the bacterial growth curve in a broth culture: lag, log, exponential, stationary, and death-decline phases. Understanding the growth curve is important for determining generation time and the action of antibiotics. The stationary phase is often key for the yield of economically important products like antibiotics and toxins. Key metabolic pathways include the glycolytic pathway, converting glucose to pyruvate. The TCA cycle efficiently generates ATP and intermediates for synthesizing amino acids, lipids, purines, and pyrimidines. The pentose phosphate pathway provides nucleic acid precursors and reducing power for biosynthesis. Metabolism is the sum of all chemical reactions in an organism. Catabolism is the process where organic substrates like carbohydrates are broken down to release energy, typically in the form of ATP. Anabolism is the use of energy produced during catabolism to synthesize cellular constituents like proteins, fatty acids, and nucleic acids. These biosynthetic reactions are essential for cell growth. Catabolism and biochemical reactions produce metabolic products crucial for bacterial identification. These tests include sugar fermentation, IM-Vic tests, which look at indole, methyl red, VP, and citrate utilization, as well as H2S production and urease activity. Various biochemical tests, such as sugar fermentation, indole production, H2S production, and urease activity, are visually represented here, demonstrating positive and negative results used in bacterial identification. Anabolic products of bacteria, often produced during the stationary phase, include toxins, invasive enzymes, pigments, antibiotics, bacteriocins, and vitamins.